In the class, we've been talking about the adult brain and how it's organized. Now we want to turn our attention to how the central nervous system, the CNS, is formed during development. Development in general is just considered a wondrous process, and it really is. And the development of the brain is certainly the most complexly orchestrated event that's going to take place as part of the developmental process. If we can just think for a minute, from a few cells, a human brain will develop that's capable of regulating the function of every other organ in the entire body. This is also the organ that is going to be able to generate a theory of relativity or to understand a theory of relativity, or a brain which is capable of appreciating a Bach fugue. And just as Hippocrates said, it is from the brain that all of the sorrows and the joys are experienced. And so from a few cells, this incredible structure we've been talking about is going to develop. In this lecture, we will discuss just briefly what is known about how the adult subdivisions of the brain arise and how some of the different cell types of the brain are generated during development. So let's start with early in development. Early on, about 18 days or so of gestation, proliferating cells along the future midline of the back are going to form a structure referred to as the neural plate. So this takes place along what will become the future back of the individual, this neural plate. And the neural plate really consists of a number of just undifferentiated nerve cells. Now, the neural plate cells are going to start proliferating. And what's going to happen is they're going to proliferate at the edges and fold up into a neural tube. So a tube is going to be created due to this proliferation and folding up of cells. So if we look at this process, we can see here, this is the surface of the body. This is what will become the skin later on. And the cells of the neural plate develop from this, and they are going to proliferate at the edges and fold up and eventually form a tube, which is then going to separate from this surface cellular structure and take its place internal to the back or the skin of the back. So this is a neural tube. Now, as you look at this tube, what you see in the middle is a hole, and that's going to be critical for things that we're going to be talking about in a minute, because what you have here is what your future ventricular system is going to be, those holes in your brain where cerebral spinal fluid is made, those same holes in the brain that Leonardo thought perception and cognition resided. So this formation of this tube is going to take place and is generally complete by about 4 weeks of gestation. The neural tube is going to close along its length everywhere except basically 3 places. It's going to close all along its length except for an anterior part, which is called the anterior neural pore, an area about 1/3 of the way down the neural tube, which is called the rhomboid fossa, and at the very bottom of the tube, called the posterior neural pore. So these are the only areas where the neural tube remains open for any time. And actually, the one at the posterior end is going to close very, very early. So we really won't say much about that anymore because it closes very early in development. But these other 2 areas, the anterior neural pore, which will be at the anterior end, and about 1/3 of the way down the rhomboid fossa, will remain open until later in development. Now let's look just a little bit closer at the development of this neural tube. So this is really a blown up picture of the neural tube. Here again you see that the tube is formed, that there's a central area which is basically a hole in this tube. The tube obviously is in this dimension, and it separates away from the surface, which is going to be the skin. When we look at this in cross section in this way, what we see is that there are cells that are right along this border of this area right here, which is the central cavity, which is retained in the formation of the neural tube. So we have this zone right here, and it's marked off here. And this is the zone where neurons are going to be born. So this is the zone of mitosis. Now, this zone will give rise to neurons and also to other cell types, which are going to be found in the central nervous system, called glial cells, which are non-neuronal cells but supporting cells. So these cells are going to give rise to neurons, astrocytes, and oligodendrocytes, all of which are specifically central nervous system cells. But once a cell decides it's going to be a neuron, then it develops very, very differently from these other cells, which we will learn about later, that are called supporting cells of the nervous system. Now, let's focus back on the neurons. Once a neuron is born here, or a cell is born and it decides it's going to be a neuron, it moves out into this zone. And so it's going to move out into this zone. Once it takes up its place wherever it wants to be in this zone, and this becomes very complicated when we actually look at the brain, but if we think of the spinal cord first, for example, once the neuron moves out here in the spinal cord and decides it's going to be a neuron, and it forms into a nucleus in the spinal cord, it's going to send out an axon. And on the very outside of this, you see it says the future white matter. So first cells are born, and the white matter won't develop until later. Now, in development, there are a number of what we call critical periods. And a critical period is just a really important concept to understand. A critical period is a window of time in which something must happen for normal development to take place. So, for example, the first critical period in the development of the brain is between 12 and 20 weeks of gestation. And it's in this critical period that most of the neurons are generated. And if anything interferes with that and the neurons aren't generated, then obviously the rest of development will not take place correctly. Because this critical period is a window of time, and that means the window can close. And once the window closes, then that process cannot take place again. Now, in the spinal cord, now we're just going to focus on the spinal cord now, because that'll help us understand the difference between the spinal cord and the telencephalon later on. In the spinal cord, the layer of neurons, which are near that central cavity, are going to stay near that cavity. So in the spinal cord, let's think of our neural tube like this again, and we have the hole in the middle. The neurons are generated in this mitotic zone, cell division zone right here, and then the neurons are going to migrate away, but they never migrate very far. So even in the adult brain and the spinal cord, the neurons are going to stay near that central hole or cavity, which we call the central canal in the adult spinal cord. So they never migrate away. The consequence of this will be is that if you look at an adult spinal cord, and this is a Weigert stained section through a human spinal cord, what you see right in the center is what is the remnant of that hole in the neural tube. What you see that is light in this section that looks a little bit like a butterfly are the neurons which are making up the nuclei in the spinal cord. And last of all, the darkly stained areas on the outside represent axons traveling up and down the spinal cord. So the white matter forms on the outside. So actually, in the adult brain, the organization of the spinal cord reflects what the original neural tube really looked like a lot. You had a central cavity or a canal, which we call it in the adult brain. The neurons are born and they move away, but they stay very close to this central canal area. And then the white matter develops on the outside. So this is what an adult spinal cord looks like. Now, in the brainstem, remember what that is. It's the older, phylogenetically older areas of the brain that the rest of the brain sits upon. In the brainstem, this pattern pretty much stays the same. You see cells don't migrate very far away from where they're born. So you see a very similar pattern. But now, in the telencephalon, it's very different. In the telencephalon, here's what's going to happen. Here's our neural tube again. Here's our central cavity. We have cells born in that mitotic area that are going to become neurons. And some of them are going to stay near that central canal area, which is going to develop into the ventricles, those holes in the brain. But some of the neurons, which are born, are going to migrate out beyond the level of the white matter to form the cortex of the hemispheres. So in the telencephalon, what you have are some neurons staying close to the central canal area that's going to develop into your ventricular system. And then white matter is going to develop because it's the axons. And then what you're going to have is some of the neurons are going to migrate to the very outside. And that's what the last two lectures were about, are the neurons that migrated out to the very outside of the telencephalon, which are the cortex. So if we look through the telencephalon and we were to cut, and this is a section cut in this plane here. So this is the front of the brain here. This is the back of the brain. So in this slice, it's stained in such a way that the white matter is white and neuronal cell bodies, so the gray matter is darker. And what you see, here's your ventricular system, and notice how large it is. It's huge in the telencephalon. This is a hole on one side, part of the ventricles. This is another part of the ventricles. And down here is another part of the ventricular system. Now, notice, right around the ventricles, the darker areas are the cellular nuclear groups that never moved very far away from the ventricles. So we have a few nuclear groups in the telencephalon that never move away from that central canal area, from that central cavity. Now, look at these on the outside here. These are all neurons that were born near the ventricle that moved to the outside and became the cortex of the telencephalon. So this occurs only in the telencephalon where you have the cortex. And this in itself, I want you to think about this. A neuron is born. The neuron has to know whether it's going to form one of the nuclei near that ventricular system or whether it's supposed to migrate a long distance to go to the outside of the brain, the outside of the developing telencephalon, and differentiate into this incredible sheet of neurons, which is responsible for all of the higher functions that we have. Now, another thing I'd like to point out and have you look at is the difference in this ventricular system, what occurs here. So here's our telencephalon up here. The ventricular system is shown internally here. If you were to look down in the brain stem and the spinal cord, it just looks like a little tiny hole, and it just maintains, it's just like that central cavity in the neural tube. When you get up here, the huge development of the cortex and the telencephalon pulls this ventricular system and enlarges it into this large hole that is found up here in the telencephalon. And because the brain develops in this C shape, like this, the ventricular system also develops into the C shape. And again, this ventricular system is where Leonardo da Vinci thought that the spirit of perception and cognition was located. We now know that these are the structures in the brain in which cerebral spinal fluid is made. Now, after neurons have migrated, their axons are going to grow out and contact other neurons. So the neurons are born, And then they migrate to wherever they're going to go, whether they're going to stay close and form a nucleus that's closer to that ventricular system, or they're going to migrate to the outside. They're going to grow out and axons and establish contact with other neurons. Now, this is incredible. They know where to go. Think about this. There's more than 100 billion neurons growing their axons in the developing brain. And each of those axons is going to make very specific synaptic connections or connect with very specific other areas of the brain. And these little guys seem to know where to go. Nothing in the brain is random. So, the first neurons, which are going to differentiate fully. So these baby neurons are going to migrate. And they're going to differentiate into whatever kind of neuron they're going to be. So there's about 150 different kinds of neurons. So for example, a little baby neuron is born, migrates to motor cortex, and then undergoes a further differentiation only in layers 5 and 6 and decides it's going to be a motor neuron that's going to extend its axon all the way down to the spinal cord and allow for the initiation of movement. Other neurons migrate to other places and project their axons. So the first cells, which are going to extend axons and fully differentiate and become neurons, are long axon neurons. And remember, the long axon neurons are going to make up associational pathways that connect different parts in each hemisphere, commissural pathways which cross between the hemispheres, connecting homotopic parts in both cortices, and the projection neurons which go up and down or connect various areas of the brain. These are all long axon neurons, and they're the first ones to differentiate. So if we look back at our drawing that we've seen before, you see our motor neuron is in motor cortex. He's migrated, he's been born, he migrated. Now he extends his axon down through the brain, crosses in the lower medulla, enters the spinal cord, and enters, say, at cervical levels to make a connection with another neuron that's going to go out that will allow me to move my arm. So I will initiate the movement on this cortex on this side, and I will move my arm on that side because those motor neurons sent their axons to the appropriate neurons in the spinal cord. Nothing is random. And you have 100 billion, actually have more than 100 billion neurons doing this throughout the brain during development. Now, the last neurons to differentiate, and this is an area, maybe it doesn't sound quite as interesting or something, but in fact, this is incredibly important. The last neurons in the brain that will differentiate and become fully functioning adult neurons are little short axon neurons. So what are little short axon neurons? Well, guys, that's where the business is really taking place. So let me tell you what they are. Let's imagine that you have a neuron that projects to another structure. That neuron, when it projects to another structure, makes connection with another structure, is more likely than not going to synapse upon one of these little tiny short axon cells that's located in that structure. Short axon cells have their cell body and their connections with other neurons only within that nucleus. So they never project their axons out. Their axons are short, they're little. So here's what you have. You have one area of the brain projecting to another, connecting with cells in that nucleus that differentiate into these little short axon cells. Then those little short axon cells connect with the neuron in that structure, which is going to project out. What that means is that little interneuron, which is only in that nucleus or area, is intercalated between input and output, which means it's the one that's doing all the integration of all the inputs that are coming into that nucleus or structure. And we are learning in modern neuroscience that there are specific disorders that involve the failure of short axon neurons to form in the brain. And one of the major disorders that we think may have something to do with such a deficit is schizophrenia. And so if we are able to, we might come back and talk a little bit about that. But these short axon neurons are the last neurons to differentiate and are very, very important in the brain. We call these interneurons because they're intercalated between input and output to a particular area. Now, just to impress you, all of these things are going on in the brain by different neurons at different times, and it's all being orchestrated genetically. It is estimated that 250,000 new brain cells a minute are being born in the human embryo. And that would impress anyone, I'm absolutely certain. Now, the second critical period in development occurs between the third trimester and 2 years of age. And this period is the time when axons are growing out for these long axon pathways, and they're making the connections to other structures. So this is an important critical period as well. This makes it all the more important if we intervene, for example, in a country where people are starving and don't have much food. This is one of the important reasons why pregnant women and very young children are the targets. Obviously, for humanitarian reasons, you want to feed everyone. But obviously, this is when the brain needs to have fuel and food. And so that is why pregnant women and very young children are targeted. Up until about 2 years of age, the brain is undergoing incredible growth. Now, I want to stop just for a second, and I want you to just try to imagine the dynamics of what's happening. You've got neurons being born, so there's birth of neurons. You've got birth of other kinds of cells from the same progenitor cells that are going to become a completely different type of cell in the brain. You've got migration. You've got differentiation into one kind of neuron or another. You've got axons growing out to other nuclei appropriately, and you're making connections with neurons, specific neurons, in these structures in very specific ways. And all of this is going on in development. Now, so how do the adult subdivisions of the brain arise? What do we know about that? That was one of the questions that we wanted to address here. Well, it turns out that the generation and migration of neurons is significantly greater at the cephalic end or the head end of the neural tube than the posterior end. So what we have is that at the head end of the neural tube, so let's imagine our tube again along the back, at the head end of it, what we call the cephalic end, it starts making neurons or being born at a higher rate, and they're migrating at a higher rate. And that part of the neural tube is going to form 3 vesicles called the prosencephalon, the mesencephalon, and the rhombencephalon. These are the 3 primitive vesicles of the brain that are going to develop into the 5 subdivisions of the adult brain. So here we show that the prosencephalon, which is one of these cephalic parts of the neural tube, is going to develop into the forebrain, which differentiates into the telencephalon and the diencephalon. The midbrain is going to become the midbrain or mesencephalon in the adult, not a lot of change there. And the rhombencephalon is going to develop into the hindbrain and going to differentiate into the metencephalon and the myelencephalon. So if we look at this in terms of our neural tube, here's what we see. The head end, which is this end of the neural tube, the head end starts making lots of neurons, and the neurons are migrating, and this is all going on at an incredible pace. And the prosencephalon is the part that's really going to develop. So these are the hemispheres here that are going to develop, and also the diencephalon. And this shouldn't surprise you, because a huge part of the diencephalon is the thalamus, which connects to the cortex, which is telencephalic. So these develop together. The other thing I want you to notice here is that a part of the eye, which we will learn is called the neural retina, actually is an outgrowth of the diencephalon of the brain. The mesencephalon in development becomes the mesencephalon in the adult. The rhombencephalon is going to differentiate into the metencephalon, cerebellum and pons, and the myelencephalon. So this is how these different structures develop. Now, if we look here at the development of the brain in cross sections, we see what is going on. This is at about 4 weeks of development. Here's the posterior part of the neural tube, and now the head end starts to develop extraordinarily. By only a few weeks later, you can see this tremendous development of the telencephalon and diencephalon. The telencephalon is going to grow so large that it's going to come over that anterior neural pore and close it off. So that's how that anterior neural pore gets closed off. In the back here, we have the mesencephalon. We also have these other areas back here. This little dark area right here is that rhomboid fossa. And you remember that that's one of the normal places along the neural tube that does not close early in development. And what's going to happen is the metencephalon, when it develops into the pons and cerebellum, the cerebellum will grow over the rhomboid fossa, effectively closing it off. And that's why the entire internal part of the brain is an entirely closed system. And by only 4 months, you can see this C-shaped structure of the brain. The telencephalon has closed over the anterior neural pore. The cerebellum has closed over the rhomboid fossa. And by birth, the brain is convoluted, it's C-shaped, and now the hemispheres are so large that you can hardly see any of the brainstem from this external surface. So the hemispheres have grown so large over this brainstem that now you can't see it from the external surface. Now, after birth, a huge number of what we refer to as regressive events occur. So regressive events mean that a huge number of neurons die. So in some nuclei and some areas of the brain, up to 70% of the neurons actually die. Many of the connections that are formed in development will also die back. So after birth, there are regressive events which take place. And even at that, it sort of fine-tunes the system. Even at that, the adult brain, we are left with 100 billion neurons in the adult brain. Now, let's think about what might happen if something goes wrong in development. It doesn't take much imagination to realize that devastating consequences occur if any part of the developmental sequence fails to happen normally. So I've just chosen 2 examples, one which is devastating and one not as devastating generally. This shows a baby who has been born, and there's his little feet and his back. You're basically looking down on the midbrain. In this baby, whose neural tube failed to close in the rostral portion, where it should have closed before 4 weeks, it did not close. And because that rostral portion did not close normally, then the whole rest of the brain never developed. The baby was born alive because the brainstem controls breathing and heart rate and all these things. And so the baby had the midbrain down, but none of the other structures of the brain developed. This is called anencephaly. And what you have, you also notice, is that there's a developmental sequence. If these areas of the brain fail to develop, then the skull and the scalp fail to develop. And so what you have is the system open, and this is not compatible with life. Now, let's imagine if something goes awry later on. And here again, we have this example. The corpus callosum may fail to develop. So one of these long axon systems is 300 million fibers. But just like the case where we went in when someone had epilepsy and cut the corpus callosum, the child may have a nearly normal life. Now, this child died not because it didn't have a corpus callosum. So you notice here, the hemispheres aren't connected. There's just an opening there. This child did not die because it failed to develop a corpus callosum. The child died because he had a number of other developmental abnormalities, and that's very common. If one thing is found to be abnormal in development, it's not uncommon to find many other things that are abnormal. So basically, development of the brain is just an incredible process. And we are born with more neurons than we will ever have again. And shortly after birth, all these regressive events take place, but nonetheless, we're left in the adult with 100 billion neurons. And that is actually, interesting enough, the adult brain then has approximately, a single brain has approximately the same number of neurons as there are estimated to be galaxies in the universe. Thank you.